Hey guys, today we're going to be doing some genetics practice problems as promised. We're going to go through monohybrids, dihybrids, codominant scenarios, incomplete dominance, sex-linked traits, as well as pedigrees. So I've already started working on a couple of these. I literally just downloaded a couple worksheets from the internet, just typed in Mendelian genetics, and this is what came up. I'm not going to solve everything for you, but just give you an idea of how you should be solving these types of questions. So the first ones we're going to look at are monohybrids. So I've already done the first one, so we'll move on to the second one about flowers. So this is telling you that homozygous, so it means the same, homo means same, red flowered plant has big R as its genotype, is crossed with a homozygous, again, same, white plant that is little r. So it wants to know what are the genotypes and phenotypes of the offspring and the probability of having white flowers and why. Okay, so it already tells you the genotypes of the parents. So these are like the easiest types of questions. So literally you're just going to plug and chug these two genotypes in our Punnett square. So you'll put parent 1 up here and you'll put parent 2 over here. It doesn't matter if you switch them. And then convention, you always put the capital letter first, like that. So you can see that all the possible the genotypes are going to be the same. So the genotypes is referring to the letters. So inside of the box, we have, 4 separate boxes, and we have 4 that are all identical. So we're going to put 100%, 4 out of 4. So 100% of the genotypes are big R, little r. And then the phenotypes is what it looks like. So because we have a dominant trait in all of these, we know that 100% of them are going to show the dominant trait. And in this case, we know that the dominant trait here is a red flower. So we know that the phenotype is going to be 100% red flowers. So it wants to know the answer to the question, what is the probability of having white flowers and why? So it is 0% chance of white flowers. And that's because you have only dominant traits in one of the parents. Basically, that's saying that this parent here is going to override whatever exists over here, because these dominant traits, if they're present, that's what you're going to see. So these monohybrids are really easy. I'm just going to do another one, not talking through it, so you can just follow along. And then I'm going to move on to dihybrids, which are a little bit more complicated. Okay, so we have hitchhiker's thumb and hitchhiker's thumb. They're expecting a child. Great. So those are your possibilities. So for genotypes, make sure that you put all of the possibilities. And make sure that the line adds up to 100. So that works out. 25 plus 50 plus 25 is 100. The phenotypes, it's asking you whether they're going to have hitchhiker's thumb or not. So we know that 75% of them will have hitchhiker's thumb. And we know that 25% will not. So that's going to be like a normal thumb. We know that because 3 of the boxes have at least one capital letter. So that's our 75%. And then our remainder here that does not have any dominance is going to be the 25%. So it wants to know what's the probability that the child will also have hitchhiker's thumb or no hitchhiker's thumb. That's exactly the same thing as the phenotype. So your answer is right there. Okay, so let's go look at the more complicated questions. Talking about dihybrids. I actually cut this first question out and I put it down on the next page. There you go. That way I can kind of show you how I do these. Your teachers will tell you to use FOIL, and FOIL is a math term, and I do not have a math-inclined brain typically. So I'm going to show you the way that I do it, because if you do it this way and you learn it this way, you'll never mess these up, and it might seem a little bit tedious, but we'll get through it. Okay. So here's the first question. It says a man has a genotype of this. So dihybrid crosses are looking at 2 different traits. So you will see 4 letters because you need 2 alleles for each gene. So 2 alleles for gene 1 and 2 alleles for gene 2. That makes 4 alleles total for having 2 traits. So he has brown hair and free earlobes. And we know that these 2 are the dominant traits because he's dominant for both of those. So then it wants to know what are the different combinations of his gametes. So you have to find that by doing that FOIL thing. But like I said, I don't have a math inclined brain, so I'm just going to do it the way that I like to do it. So there's just a convention. It's something you just have to memorize. I'm just going to write his genotype down here. We have big B with little b and big E with big E. Okay, that's his genotype. So basically, do you agree here that we have 1, 2, 3, and 4 letters? Makes sense. There are 4 letters. We're dealing with 2 different traits, 2 alleles for each trait. 2 times 2, 4. Beautiful. Okay, so there's just a convention, something you need to memorize. It goes 13, 14, 23, and 24. Essentially, this is FOIL for dummies because apparently that's me. Okay, so in position number 1, what do we have? In position number 1, we have a capital B. So we're going to write that. In position number 3, right there, we have a capital E. So that is our first gametic combination. The next one down, 1 with 4. So we're literally just like doing this. Okay, so we're going to have a big B with a big E again. It's the same, but that's okay. Okay, 2 with 3. So now we're going to distribute here. So I guess it's like a distributive property. Again, I am not good at math. This is how I like to do this. So these are going to be our gametic combinations for dad. Okay, so that's what it wanted to know up here. So in this little blank, you would write that you have big B, big E. You'd have big B, big E. You'd have little b, big E, and little b, big E. So those are the combinations for dad. Okay, then this man met a woman, and her genotype is this. So we're going to do the same thing for her. Okay, so she's big B, little e, little e. I'm going to write that over here. Okay, so again, we have 1, 2, 3, and 4 letters. The standard confirmation here is going to be like this: 13, 14, 23, 24. And again, that's just distributing these letters like this. For me, I don't want to draw these each time. I just remember this order and it works. So again, position 1, we have a big B with a little e. And then 14, we're going to have a big B with a little e. 23, coincidentally, is the same. And so is 24. Very convenient. So really, she's going to make these different gametes, but they're all going to have the same genotype, which is totally okay. That just has to deal with her genetics. So these are her possible gametes that she can make based on these 2 traits. So then essentially you're going to take this information from dad and this information from mom, and you're going to plug them in. Oops, I switched them. It doesn't matter. Mom goes here, and dad is going to go up here. Okay, so we'll just write out those combinations. Notice that I always kept them in alphabetical order. That's something that's important. Typically, like I said, you want to put the capital letter first and then the lowercase letter will come next. But when it's like this combination, you still have to keep them in alphabetical order. Okay, so we're going to put dad up top. So these are his possible gametes that he has. Okay, and then all we have to do is go through and fill out the squares. Okay, so you're always going to have your B's touching and your E's touching. No matter what your letters are, you keep them together. So you do not do this where you do like B, E. That's not okay. That we're not talking about the BB, Bebe, whatever. I'm not cool enough to know that brand, okay? So you're going to keep the B's together and the E's together and make sure that you always put the capital first. So that's what the first square would look like. The second square, you're going to have big B with big B. You're going to have big E with little e. Your next square, you're going to have big B, little b, big E, little e. And then the next square is actually going to be exactly the same. And this one worked out really nicely because if you look, all of these are the same. So all of these are also going to be the same. So really, you only had to solve like right here to get your answers. Okay, so that's how you do a dihybrid. I am going to draw out the hardest dihybrid for you. It's not like an example from the paper. It's just one that I'm going to draw out for you because once you do that, then the rest are pretty simple. Okay, so the most complicated one is if you have a double heterozygous cross. So if your cross looks like this, that's the most complicated one that you can get because you're going to have literally a whole bunch of different genetic combinations at the end. So all we're going to do is the same thing with our 13, 14, 23, 24. So in position 1, 2, 3, 4, we have a big A and a big B in 3, and in 1 we have a big A, and in 4 we have a little b, and then 2 is a little a, 3 is a big B, 2 is a little a, and 4 is a little b. So these are the possible gametes for one of the parents. Okay, then the other parent, what do you notice about them? Hopefully you can see that they are the same. So their gametes are going to be the same combinations, and they will look like this. So then if we want to put those on a Punnett square, I'll just draw it out. An easy way to draw these, if you have to, is to draw a big box. You divide it in half, and then you divide it in half again. And then the other direction, you do the same thing. So divide the box in half, divide it in half again and again. And then you have the gametes that you're just going to add on to the boxes. And it doesn't matter if mom or dad goes on the top or the side. It doesn't matter. It's going to give you the same result. Okay, so I'm just filling these in for you. And then I'm going to go through and solve it. So again, you're going to put your A's with your A's and your B's with your B's. So our two big A's are going to touch, and then our big B's are going to touch here. And then I'm just going to go down. So big A, big B, little b. That's how you'd solve this whole, can't see that. That's how you'd solve this whole square. So you just keep going. And here we have big A, big B, little b. You have big A, little b, big A, big B, little b, big A, little b, little b. So you just keep going to fill out the entire square. Like I said, this one has the most genetic combinations, so it's going to be a little bit tedious to do like the next step, but we'll get through it. So then this one over here is our last column and our last box. Okay, so now that we've solved our square, right, we're going to do the genotypes. And you're thinking like there's a heck load of them, and you're right, there are. Okay, so I like to think about the squares as being like number 1, 2, 3, 4, 5, 6, 7, 8, all the way down to 16, because there are 16 squares. Okay, I think that that's the easiest. I like to start in square 1. So what is the genotype present there? You have big A, big B, big B. And how many of those do you have out of 16? You actually only have 1 out of 16. So that's accounted for. Square 2, you have a, big B, little b. You have 1 here, and you have 1 here. So that is 2 out of 16. Next, we have big A, little a with a big B. You have 1 here, you have 1 here. So that is 2 out of 16. In box 4, you have big A, big B, little b. You have 1, 2, 3, 4 of those. That is 4 out of 16. Then we did 5 already, so 6 is big A, little b. You actually only have 1 of those. How do I know this? Because I've done this for a long time, but you can also like go through and check each of the 16 boxes if you don't believe me. In box 8, we have big A, little b. We have 1 of those here and 1 of those here. Notice that they're always on diagonals, which is really convenient for finding them. Next, we have little a, big B. You have 1 here. That is 1 out of 16. Then you have little a, big B, little b. You have 1 and 2 of those. So there are 2 out of 16. And then you have our all recessive traits here, which is 1 out of 16. Then to make sure that you've finished, you just add them up. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So 16 out of 16 means that we did it correctly. The next part would be to do the phenotype. I didn't give you any parameters for that, but you can still identify what they are. So you know that you can always have a dominant trait A, right? You can have dominant A with dominant B. So I'm just going to say dom A and dom B. That's one combination. You can also have dominant A with what's the alternative? With recessive B. So A's been with big B and little b. So then the flip side would be that you have a dominant B with a recessive A, or you have a recessive A and a recessive B. So those are the possible combinations. So let's go through. We're going to use the genotype actually. So if you have a dominant A, you know that you're looking for like an A, and then it doesn't matter what's next to it. And if you're looking for a dominant B, you're going to be looking for a big B, and it doesn't matter what's here because if it's dominant, it's always going to show. So we have this one works for that, this works for that, this works for that, and so does this. So looking at this top section, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 here. So 9 out of 16 are going to be dominant for both traits. Next, we have dominant A, which means we're looking for A with who cares what next to it, and then recessive B, which has to be little b, little b. So big A with little b's. Big A, little b's, big A, little b's. Looks good to me. 1, 2, 3. We have 3 of those. So 3 out of 16 have a dominant for trait A and a recessive for trait B. The next one is actually just flipped. So dominant B, you're going to have a B and who cares, and then recessive A's. A always comes first, so I wrote this backwards. Sorry. We're going to have little A's with the big B, little A's with the big B. That looks good to me. There's 3 of those. So you'll have 3 out of 16 there as well, which is really, these two are just inverses of each other. And then recessive everything is going to be all little letters, and we happen to have 1 of those left, 1 out of 16. So if you look at this ratio, it's called a 9-3-3-1, and it's special for when you have a double heterozygous cross like this. It's a double heterozygous cross. Everything is heterozygous. This is always going to be the phenotypic ratio for these kinds of crosses. And if you can just memorize this, it will save you a lot of, doing all of this. Okay, so that's a little bit about our dihybrid crosses. So we're going to talk about codominance and incomplete dominance. So I like to just explain that for codominance equals both show. So whatever the parents are in the offspring, you're going to see both traits. Okay, and then for incomplete, I like to think in between. It's like a mix or a blend. Okay, you can't see either of the parents, but the offspring are a combination. So let's say that you have like a white flower and a red flower and it's incomplete dominance. Well, then white and red make pink. Can you see white and red? No, but you see a blend of them that's incomplete dominance. Okay, so we're going to practice a few of these. So this tells you that in a certain fish, blue scales, this genotype, and red scales, that genotype, are codominant. Actually, you would have 2 different letters. So that's actually the wrong convention. So let's just fix that for them. You'd have capital R's because they're both dominant. So when a fish has the hybrid genotype, it has a patchwork of both blue and red scales because it's codominant, so both show. So we're going to use the letter B and the letter R. So this is, like I said, actually incorrect. So we're going to go through and solve that. So if you have these 2 parents, I'm going to use this square right over here. If you have a big B, and you have a big R and a big R, then this is what all of the offspring could possibly look like. And it tells us that the hybrid or the heterozygous or the one that has both traits is going to be a patchwork. So it's going to have a mixture of both blue and red scales. So that's what's happening here. I'm actually not going to go through and solve the rest of it for you. Like I said, the convention with their little b's is actually incorrect. So now it wants us to take 2 of these and put them here. So let's do that. So we're taking 2 fish that have both blue and red scales, blue and red, and we're going to mix them together and determine the probability of their kids looking like them or looking different. So in this case, you have 25% of these that are going to be the big B. Those are going to have blue scales only. The fish that have, there's 50% here that are big B and big R, those are going to be both blue and red. They are not purple. They are blue and red because co means both show. And then lastly, we have big R. There are 25% of them, 1 out of 4, which means that they will have red scales only. So that's what those look like. Okay, and basically, this is all wrong. Oops. This is all wrong. It has nothing to do with codominance, actually. So we're going to move down to the next part. This is incomplete dominance. So incomplete dominance, remember, is in between. Also, I forgot to mention, so for codominance, back up here, both show. Hello. Both show. This is something that was discovered in chickens. So they like to use the speckled chicken a whole lot. So you'll have like a white hen and a black rooster. And when they have babies, they're speckled black and white. Think like Dalmatian chicken. Okay, that's essentially what happens. Is that how Dalmatians are bred? No, but that's how these speckled chickens are made. Okay, because that feather pattern has codominance. So incomplete is going to be in between. So we're going to see a mix or a blend. Okay, so in snapdragons, this again is where this was discovered. That's why you always hear about snapdragons. It's a flower. So flower color is controlled by incomplete dominance. There are two different alleles, red and white. I don't know why they do this like this. This is still wrong. It's codominant, so both letters are capital, and you do it like this. This is just wrong. Okay. So the heterozygous is going to be pink because a blend of white and red is, pink. Okay, so the phenotype of a plant with the genotype big R, well, that's going to be red. And what they mean here in the next one is big R, big W, which would be pink. And then if you had WW instead of this jazz, you're going to have white. So that's what it really wants you to know. So the whole point of this is that they're both dominant. You would not have a recessive looking letter. That's all kinds of jacked up. Anyway, you're going to have a pink flower plant crossed with a white flowered plant. So the correct way to write pink equals this because it's the mix. And a white flowered plant would be this. Okay, what's the probability of producing pink flowered plants? Well, let's find out. So RW and WW. So we have these two are going to be pink. So we have 50% chance of pink flowers, and we have a 50% chance of what? Those are white. Okay. So let's do another one. So what cross would you produce with, will produce the most pink flowered plants? Show Punnett square. Okay, so we want the most pink flowered plants. And what's pink? It's this and it's this, which means that this came from one parent and this came from another. So I think that this is probably the best way to go about it. Let's find out. So if you have a red flower and a white flower and they mix, this is what you will get at the end. Look at that, 100% pink. We did it. Fantastic. Okay. The next one, obviously, if this is white, you would not have this. You'd have a big W and a big W. That's not a W. I got excited. That's a tri-U. Anyway, you can go ahead and keep practicing these on your own if you wish to. Like I said, I literally typed in incomplete dominance practice sheet, and this is the first thing that popped up. And as you can see, it's wrong. So you can't trust everything on the internet. Anyway, one of the best examples of codominance is blood typing. So these are the different blood types here, the phenotypes, and these are the possible genotypes. I also just write them out like instead of like I to the A, I do just like AB is like this. O is always like this because you can tell that I's are I's and they're not zeros. And then for A, it can be either big A, or it can be big A, I. And then here you can have big B, or big B, I. If you raise it to like I to the B, that's fine. It's talking about the antigen that's present on the outside of your red blood cells. That's what makes your blood type. So you have red blood cells. And if you have like these little like spiky guys, like maybe that's antigen A. So this is type A blood. And if you have type B blood, maybe you've got like more of like these like little like speckles, circle-y looking things here. And if you have type O blood, you have nothing on the outside. And if you're type AB, then you would have both the spikes and the little speckly domes, right? I'm so good at drawing. This should be my next career. Anyway. So those are the possible blood types, and that's what that means. And then if you've heard like, oh, I'm O negative or I'm AB positive, the positive and negative is something called the RH factor, and that's not something that we're talking about for codominance. So you don't really need to worry about that. Okay. But this is literally what the blood type is referring to. It's the antigen on the outside of your red blood cells. Okay. So it wants to know here the homozygous homozygous genotype for the B allele. So if it's homo, then it's literally going to be like that. If you're looking for heterozygous A, it would look like that, which means you have one A trait and one O trait. So half your blood is like this and half of it is like this, which is totally okay because O can go with anyone. Type O is little I. That's it. Type A and a type O parent, which means you'd have to have type A and then an I because your parent was type O. They can only give you an I, so you're a heterozygous. Type AB is only like this. Blood that can be donated to anybody is type O because it doesn't have any antigens on it, so it's not going to cause a clot and kill them. And can only get blood from type O. Donor is going to be our type O because O can only get O, which kind of sucks. Like AB is like anyone. It's a universal receiver. Okay, so let's look at an example here. So pretend that Drake is homozygous. Homo means same. Homozygous for type B. Okay, homozygous for B is going to look like this. And Nicki Minaj is type O. Well, I obviously didn't create that because I don't like these people, but that's fine. So let's show our work on the square here. So this is going to be the probability of their offspring having these particular blood types. So you can see that we have 100% type B. And they're all heterozygous, okay? So draw a Punnett square showing all the possible blood types for the offspring produced by a type O mother, which is little I, and a type AB father. So A and B, that's the easy one. A and B with type I looks like this, or type O, sorry, looks like this. So you can have type A, you can have type A, you can have type B, or you can have type B. So you have 50-50, so 50% A and 50% B there. That's our codominance, and it's codominance because co means both show, and as I drew in my beautiful diagram here, both antigens are showing on the heterozygous, the one that is mixed. That's why it's codominance. Okay. You can continue on to practice some of these if you'd like. I am going to continue on to show... Hello? Pedigrees. Okay, so pedigrees, the circles are females, the squares are males. If they're colored in, it means that they have the trait. Sometimes they like to do a half-Z's, which means that she is a heterozygous individual. They don't always have to do that because if they're a heterozygous individual and it's a recessive trait, they're not actually affected by the trait. So the easiest way to do this is to just start looking at one. So here's an example here. So a male, it just wants us to list any male. Okay, so a male is going to be a square. So let's put down C. A female is going to be a circle, so A. And a marriage is going to be B. A marriage is going to be a horizontal line, whereas offspring are going to be like a vertical line. So a person who expresses the trait is going to be A. They are always colored in. A person who does not express the trait is going to be C because they are not colored in. A connection between the parents and the offspring is going to be D. It's showing you this vertical line that this is mom and dad, and then they had these children. They had a daughter, they had a son, and actually she's the oldest and he's the youngest. That's how that works. How many generations are on the chart? You start at the top. This is generation 1, 2, that's a 3, Kate. Okay. 1, 2, and 3. There are 3 generations on this chart. Okay, so assuming the chart above is tracing a dominant trait for a white forelock, which just means it's like a little tuft of hair on your forehead, what is the most likely genotype for individual A? Individual A is affected. If it is a dominant trait and... Okay, I see what they're trying to get you to do. Okay, it's a dominant trait, so we know that it can't be this, right? So if you look at the offspring, there's a 50-50 chance that they're going to get it. You notice that the dad does not have the trait, so the dad has to be little f, little f. So we're trying to figure out if we have a big F situation here or a big F, little f. Okay, but because half of the kids have it and half of them don't, it would have to be the heterozygous. So let's look at that, okay? If you have big F, like if that's what we're working with for mom, we know that dad is little f, little f. We already know that because he doesn't have it. That means that every single one of the kids, 100%, right, is going to have the dominant phenotype. And that's not what's happening, because up here you can see that this daughter and this son are not affected. So you know that you have to have the heterozygous for mom, because she's going to be big F, little f, and dad we already know is little f, little f. Well, here is the combinations. Then you have a 50% chance of them having the trait and 50% of them not, and that's exactly what we see here. So that's how you know that this individual has to be heterozygous. For individual C, well, we just discovered that because it's a dominant trait. If it's not colored in, well, they have to be the recessive trait. So this is dad in that situation. Okay. So we'll look at this example. So it wants to know what Arlene's genotype is. So Arlene is here. She's colored in. Okay, so the carriers in this are not indicated with half circles. Okay, so what are we dealing with? This is a dominant is normal, recessive is the anemia. So she has to be recessive. She has to be, because she has the trait. Oh, it says to use the letter F, my bad. She has to be recessive because she has the trait. And up here in the question, it tells you that it is a recessive trait called anemia. Okay, so it wants to know George's genotype. Well, this is exactly the same thing that we just did. Because 2 of the children have the disorder and 2 of them do not, we know that George is going to have to be a heterozygous individual. Otherwise, you could not get those combinations. So he's going to be big F, little f. Okay, Anne and Michael's genotypes. Where are Anne and Michael? Anne and Michael are over here, and their daughter has the disease, but they don't, which means that both of them are carriers. So they're both going to be big F, little f, and big F, little f. And if you don't believe me, here's the Punnett square real fast. Okay, so it looks like this. You have this individual right here as having the disorder. So they both have to be big F, little f, and it asked for them together because they're going to have the same genotype. Okay, so that's basically how you do our pedigrees. I only have a few minutes left before my kiddos come in, so I'm going to move on to sex-linked traits. Pedigrees are pretty simple. So sex-linked traits are traits that are linked to a sex chromosome, and know that by convention, females are XX and males are XY. Okay, those are the only 2 options. I know it's 2020, but listen, biologically speaking, these are the only 2 combinations of sex chromosomes that you can have, and then also reproduce offspring. And we're studying genetics, which is carrying on your traits from one generation to the next. So, that's what we're dealing with. Basically, most of the sex-linked traits we're going to talk about are linked to the X chromosome, which is actually an advantage for females and a disadvantage for males, because that means that males are more likely to get the trait because they only have one copy of an X. So if their copy of X has the trait, they're going to have it. For females, if they have one copy that has the trait and one copy that doesn't, then they're just a carrier. If both of their X's are affected, then they actually display the trait. Okay. I don't really want to read all this out. I'd rather just do a practice. Okay. So in fruit flies, the gene for white eyes is sex-linked recessive. Okay, so this means it's going to go on the X chromosome and it's going to be recessive. So big R is red and little r is white. So cross a white-eyed female. Okay, so a white-eyed female. A female, I'm going to do it down here. A female is XX and she has white eyes. And it says that white eyes is recessive. So this is going to be her. And the man, fly, he has red eyes. Red is going to be a capital R, and I know that because it says it right there. The Y does not get anything because it is not linked to that sex chromosome. So these are going to be what we put on the chart. So we're going to have X to the little r, X to the little r, X to the big R, and Y. Then you just go through and you solve. And yes, this is the correct order to put everything in. You still put your capitals first. Okay, so this is your overall result. What percentage of the males will have red eyes? Red eyes is dominant. So these 2 are males right here. Neither one of them is dominant. So 0% will have red eyes. What about white eyes? Well, this is the white-eye gene, and this is the white-eye gene. 100% of males will have the white-eye trait. Okay, what percentage of females? It's going to erase my boxes. Yep, dang it. Okay. Hello, this is like this, and this is like this. Let me just do it again real fast. Okay. Percentage of females have red eyes. Red eyes is capital. So this one and this one both have red eyes. So 100% of the females have red eyes. White eyes has to be 0% if you already have 100. Okay. What total percent of the offspring will be white-eyed? Total percent of the offspring. So white eyes is recessive. So 2 out of 4 is going to be the white-eyed trait. So 2 out of 4 gives us 50% of the total percent of the offspring. Okay. So notice here that these females have red eyes and not white eyes. That's because it is a dominant trait to have the red eyes and a recessive trait to have the white eyes. So you would need 2 copies of the X's that have just like your normal genetics, right? So that still works the same. So these 2 females are actually going to be carriers for the trait, but they're not going to have it themselves. They can pass it on, but they will not be affected. Okay, let's try this one. So using the same information as question 1, cross a heterozygous red-eyed female. So female is XX. And if she's heterozygous, she's going to be big R and little r. And red is dominant, so that works out. With a red-eyed male. Okay, a male is going to be XY. And if he has red eyes, then he has this genotype. Genotypes of the parents are this. And then we'll go ahead and solve. So x to the big R, x to the little r, x to the big R, and y. Okay, so what fraction of the children will have red eyes? Okay, well, anything with a capital letter. 1, 2, 3. 3 out of 4 will have red eyes. What fraction will have white eyes? This one right here. 1 out of 4. Okay, what fraction of the female children will carry the white-eyed trait? This one right here, because she has this white-eyed trait right here. So it is 1 out of the 2 possible females. A lot of you probably would have put 25%, but that is 50%. That is 1/2. Okay. So that's how your sex-linked traits work. Real fast, I'm going to go down here and just do one more example with color blindness real fast. So again, a female is XX and a male is XY. Think about what your mother can donate to you. She only has either an X to give you or an X to give you. She can't give you anything else. So you know that every egg cell has an X in it. A male has an X and a Y, which means that he can either donate an X to you or he can donate a Y to you. So whether it is an X sperm or a Y sperm that fertilizes an egg, the sperm is actually what determines the sex of an offspring, because every egg cell on the planet already has an X in it. So really it's a sperm that's either carrying an additional X or a Y. So if you have traits that you don't like, you can, just blame your dad. Anyway, that's only for sex-linked traits. Let's talk about color blindness. So I like to show color blindness as just a slightly different convention than what we just looked at. And you'll see this in other textbooks and things too. So a normal female will be XX. A carrier female will be XY. And an affected, I just lied to you. A female is not XY. Nobody caught that? Great. Hold on. An affected female. I should have listed these out first. Okay, so a carrier female is going to look like this. She's a carrier. Whatever the trait is, she's a carrier for it. And then an affected female would look like this. So sometimes this is how you'll see it. Instead of this dominant and recessive, sometimes you'll see it like if the X has nothing on it, it's normal. And if it has a letter on it, it's not normal. Sometimes you'll see it like this. And so for males, you can have normal or affected. If you're a normal male, you'll look like this. And if you're an affected male, you'll look like this. Sometimes you'll see this convention. Okay. So notice that there's only 2 options for males. That's why they are much more likely to have a sex-related trait compared to females. Okay. So let's say that we have a colorblind dad and a carrier mom. What would that look like? Colorblind dad, he's affected, he's colorblind. And a carrier mom, she's a carrier. She has one copy that's affected. The other one is not. Is she colorblind? No, she's a carrier for it. So then let's do the Punnett square for that. Yes, sometimes I like to not draw my squares because it just takes more time. And sometimes I'm lazy, and that's okay sometimes. Okay, well, I'll draw it for those of you that like to see it. Okay, there you go. So here we have our females on this side and our males on this side. So 50% of the females are colorblind. And 50% of the females are carriers. And then with our males, 50% are colorblind. Can males be carriers? No. And then 50% of them will be normal. Okay, so that's just an example of color blindness for you. And you can actually track sex-linked traits in a pedigree. Remember that the pedigrees are these little pictures here. You'll know it's a sex-linked trait in a pedigree because you will have a high number of males that have the trait compared to females. That's how you can tell the difference between like this one is like a normal, like dominant or recessive versus a sex-linked trait, because most males will be affected whereas females will not. Okay, so I hope that makes sex-linked a little bit clearer. We went through monohybrids, dihybrids, incomplete dominance, codominance, pedigrees, and sex-linked traits. Thanks for joining me. I'll see you in the next one.